Okay, well, let's go ahead and get rolling. So, saved in the last minute with the projector. So, a couple of announcements. Quiz 4 is going to be due on Wednesday, so make sure that you get that done. You might want to wait a little bit still on the material. We're still kind of sludging our way through it. By the end of the day, we should cover most of it. I said last time we got together, I said that the group work would probably be on Wednesday. I'm going to make an announcement right now. I'll say it's going to be on Monday unless I don't finish the material today. Okay, so I'll make an announcement at the end of the class, say, oh, let's move it to Wednesday, or I'll say, yeah, we're going to keep it on Monday, okay? You don't really need to prepare for that necessarily. It's going to be essentially a group work on glycolysis. Okay, so someone in your group needs to have a smartphone or a computer. So know that. Okay, there's going to be a recitation tonight. Remember at 4:15 in Walnut 103. There will be another recitation on Sunday at 3 o'clock, and that's going to be MRB 312. MRB is probably going to be locked, so the TA said he would just prop the door open, so I'll tell him that I told you guys to go in the east door, okay, the door facing physiology, so that will be the door that's opened, okay? All right, and I think that's it. Why wasn't Bill put in jail after killing dozens of people? Morbid. Anyone? What's that? I hear mutterings, but no one's brave enough to answer this one.
Capital punishment?
No, not capital punishment. Yeah, you're like, Bill was killed afterwards. No, this is not. No.
Is he the president?
Huh?
Is he the president?
Is he the president? Yeah. Presidential pardon for himself. No, this is not that level of corruptness. Bill is not a person. He's it's a hurricane. That's a good one, right? Okay. Okay, so Wednesday, we left off essentially right here. We looked at, on Wednesday, if you remember, the biochemical processes to absorb glucose and get that into the bloodstream from the lumen of the small intestine, after which that blood will then dump into this portal vein, which will carry anything really coming from the small intestine that's absorbed in that manner to the liver. Okay. Blood from the pancreas, anything that's being secreted from the pancreas, is also going to go right to the liver. Big things there would be insulin and glucagon. So this is something just anatomically speaking, obviously this is macroscopic, to consider. After these things go into the liver, then they'll be distributed through the rest of the body. So what we're going to do today is we're going to start to think about then how does that glucose, those carbohydrates, in particular glucose, get into tissues, and we're going to be contrasting really today two different tissue types, the liver and the skeletal muscle. Although we'll say the liver and the pancreas are very similar in their capacity and ability to absorb glucose. So we'll kind of put them on equal playing ground, and then we'll talk about skeletal muscle. At some point in time, I'll probably mention neuronal tissue. I don't know if I'll talk about that today, but we might mention it. There's some interesting things there, okay? But to start off then this conversation, we need to just first of all consider what would be needed for any cell type to absorb glucose, and that is going to be, of course, a transporter. Glucose and all carbohydrates are polar molecules. The membrane is impermeable to them unless it has some transporter that it can get that will allow it to go through that membrane. The name of the family of transporters that does this are what's called GLUT transporters. And what the table that you're looking at here is definitely an abbreviated list of the types of GLUT transporters that you can find in our bodies and in biological organisms. Okay, I'm listing off here 4 of them. I think there's now in excess of 20 of these that have been cataloged thus far. So we're going to look at really 2 of even this 4. So we'll even cut this list down a little bit, but this is just to give you some exposure. So I have GLUT 1 through 4 here, the tissue distribution of those. Okay, Importantly, GLUT2 would be found in the liver and the pancreatic beta cell. Okay, those are the two that we're going to be thinking about in regards to the properties of GLUT2 and why they're important for those tissues. We'll be contrasting that to GLUT4, which is found in the muscle cell. It's also found in adipose tissues. Okay, so and those are the things that we're going to concentrate on. And maybe we'll get to GLUT3 neurons later on. Okay, a neuronal system. However, in all cases, okay, moving over to this portion of our slide here, over to the right, if we were to go ahead and set a concentration of glucose on the outside of a cell, and then measure the rate of uptake of that cell based off of this concentration of glucose on the outside, measure that rate and then plot it, and then set our experiment up again, have a new substrate concentration on the outside, and keep measuring that rate, increasing substrate concentration all along, okay, we would get something that looks like this. You'll notice this is VO, which is velocity. It says initial velocity of glucose entry, so glucose entry into the cell, versus, in this case, substrate concentration on the outside of the cell. Okay, and these would be cells that are equipped with these GLUT transporters. And what you see here is this type of behavior that, if you remember, going back to our discussion in chapter 1 or lecture 1, it mimics what would be considered a Michaelis-Menten type enzyme that has this hyperbolic saturation profile. In fact, that behavior, mathematically speaking, is identical. We can fit that behavior with the Michaelis-Menten equation. Okay? Now, your book, the Linninger's text, doesn't have to do this, but it tries to do this for clarity's sake. It replaces what would be the Km in the Michaelis-Menten equation with what they call the Kt. Mathematically, exactly the same concept. It is the concentration of substrate required to reach half the maximum velocity, in this case, of transport. Okay. It also is a measure of specificity of that particular GLUT transporter for carbohydrates, in particular glucose. That's what we're talking about here. Okay. And that's what we're going to be thinking about primarily today as we consider this, or these GLUT transporters, is the Kt. So coming back over here then, looking at our table, you'll notice that I have reported Kt's. These are millimolar, that's our units here, so that's a concentration. And at those concentration of glucose, those GLUT transporters will be at 50% of their maximum velocity. Okay? And you'll notice that there is differences. Okay? In particular, GLUT2 is really high relative to the others. GLUT3 is really low relative to the others. Okay? And there are good reasons for that, and we're going to talk about today. So let's start off with this, okay? Let me start you off thinking in this direction, my question, okay? Talk to your neighbor about this. I'm going to give you 2 minutes to do this, okay? Let me go ahead and expand this question. I'll pause it here, okay, but that way we can see it, okay? If the Kt or a Km of a particular transporter, okay, and we could apply this to enzymes as well, is 15 millimolar. So that's the Km, Kt value. And the substrate concentration is 5 millimolar. What is the percentage of Vmax, or what percentage of the Vmax will the transporter or enzyme be at? Think about that. Talk to your neighbor about that. How would you demonstrate that? How would you do that mathematically? Okay, you can come to an exact answer on this. Okay, so what do you think? What is the percentage there? So talk to your neighbor about that. I'm going to go ahead and give you 2 minutes. And your marks is set, go. Okay, what do you think? I will come back here and show you this slide so you can see the Michaelis-Menten equation. Okay? We have a minute left, so you can get some time. Give me a percentage. About 30 seconds left. Okay, about 10 seconds. All right, submit an answer. Submit it. Okay, I got about 87. Yeah, that's okay. All right, so let's look at these things here. Okay, so our word cloud. I hope that someone doesn't really feel that way. If you do, please leave right now. If that's your emoji, if it's the question that made you feel that way, you're really not going to throw up. But if you just feel that way when you walked in the door and you can stand up now and leave or later discreetly. What I don't want to see happen is it come down the row, right? Okay, it's going to tilt towards me. All right, so 25% looks to be the most often given answer. Let's go ahead and look into this a little bit more. Okay, 16.66666%. Okay, so very exacting on that one, 25%. The natural logarithm of 0.2. Okay, 17%, 75%, less than 20, about 20, 25, 50. I don't know. Vmax divided by 15, 15%, 15-ish. Honestly, I'm not sure. Okay, there's our throw up guy. Okay, all right, so well, the answers really are kind of all over the place, I'll be honest with you. So let's do this, okay? Let me show you, first of all, just how you can figure this out. Okay, so here's our Michaelis-Menten equation. Now, I made the numbers actually pretty easy on you, okay? The substrate concentration was 15. No, I'm sorry, the substrate concentration was 5 millimolar, and the KT or the KM was 15 millimolar. Okay, there's a mathematical difference between those, obviously. What we can say is this: the KT would be equal in this case to 3 times the substrate concentration. A way to think about this, right? So if 15 equals 3 times 5, that's what we have. From that now, we can do some pretty easy substitutions in arithmetic. All right, so we can take this and say, okay, well then VO is going to be equal to Vmax times the substrate concentration over KT, but in this case I'm going to put 3 concentration S because they're the same thing, plus concentration of S. This is going to reduce to this, Vmax times S over 4S. Cross these out, and I'm at 1/4 my Vmax, 25%. The correct answer, okay? And you say, oh, that is so useful. Thank you so much for doing that. I'm so happy now. It all makes sense, and now I see where you're going with this. Okay, so since you see that, let me ask you another question, okay? Who cares, right? I mean, that's essentially this question. How does knowing the KT of the GLUT transporter help us understand the physiology of a specific tissue? Okay, based off of the thing I just did, what's the importance of doing all that? Who cares about the KT? Why is that important? Okay, so and there is a connection here. So I'm going to give you 1:45. Talk to your neighbor about that. Why is that important? Okay, think about that. And that's going to help us with our day's worth of material. Okay, what do you think? Why is this important? It's a hard question to respond to. I get it. I understand that. Give me a response. It's just to get you thinking. Got about 10 seconds left, so submit something. Submit an answer. All right, we got less responses on that one, I know. It's okay. If you got the first one, you're going to be okay with today. Okay, so let's look at these. Oh no, I need to change this again. Save. Okay. The cell types that require higher concentration of glucose will have a higher KT. Okay. Can estimate affinity. KT, I don't know why that one's so big, but let's read it. KT is a measurement of affinity. A higher KT indicates a lower enzyme substrate affinity, so the measurement of glucose across the membrane takes longer. Okay. Helps determine the level of substrate needed to significantly change enzyme velocity and thus function of a tissue. Okay. That's an interesting one. So let's stop right there. Helps determine the levels of substrate needed to significantly change enzyme velocity and thus function of a tissue. That's about what I'm looking for. Okay. That is the reason that we care about KTs. Let me go ahead and show you this graphically. Okay. And this is why the Michaelis-Menten equation, right, and these types of things become important. Okay. So I'm going to go ahead and erase all this. Hopefully you've written all that down. There we go. Get rid of all that stuff. Okay, and I'm going to go ahead and draw the Michaelis-Menten or graph up here. Okay, so VO and substrate concentration. This is glucose. Okay, and we've got this hyperbolic saturation profile. Now, let me just use the numbers I was using last time. Okay, and let's just say that. So we've got this Vmax here. It's being approached by the transporter, and let's just say then that this happens to be half Vmax. Okay? So we drop this down and we say that, okay, so our KT is equal to 15 millimolar. Right? So that's our KT. Now here's what's interesting. Okay? Physiologically speaking, glucose concentrations in the blood is about 5 millimolar. Okay, about a third of that. Range is anywhere between 4 to 8 millimolar. Okay, that's our range. So what does that do here? Well, let's put this. So that's 15 millimolar. Let's just say, it's hard to say without a scale, but let's just say that this happens to be 4, and this happens to be 8. And go right here, drop this over, go all the way up here, drop this over. Okay, notice what happens if we're at a very low substrate concentration, glucose, right? 4 millimolar in the blood. What happens if you eat a meal and it goes all the way up to 8 millimolar? What happens to this particular GLUT transporter? It's a pretty large increase in the rate of absorption, right? And the GLUT transporter has almost a linear rate. Okay, not quite linear because this is a hyperbolic equation, right? But it's almost linear in terms of its rate of absorption. It goes from a very slow amount to a fairly high amount, not above 50, okay? But there's a significant change in this. Right? This particular GLUT transporter will be very sensitive then to changes in glucose concentration because its KT is so large. Right? Now compare that then, I'm not going to draw this graph, but compare that, let's say that the KT dropped all the way down to 1.5 millimolar. Okay, well in that case, I guess I should draw the graph. Let's go ahead and draw the graph. 1.5 millimolar. So this is going to be something like this. In that case, and you get a really small change in the rate of absorption. Okay? Because we're starting to have the physiological concentrations of glucose in this case, okay, are significantly higher, quite a bit higher than your KT. Okay, so you're starting to reach that leveling off point in the saturation curve. This is going to be really important. Okay, so coming back to this, right, they have different KTs. In particular, that GLUT2 transporter has a really large KT, which means that it's going to be sensitive. Okay, so that's going to be important as we try and understand the function of the liver or the pancreas and its ability to absorb glucose. Any questions on that? It's always nice to put reasons to why we talk about like Michaelis-Menten equations and KMs and these types of things. This is part of the reason. And be very helpful in understanding things from that standpoint. Okay? Now keep that in the back of your mind. Okay, we're going to come back to that. But let's go here. Okay, what happens once glucose is in the cell? Okay, so we just talked about glucose getting into cell. What happens when glucose goes into the cell? Okay, the thing that happens right away in virtually every cell type that will absorb glucose is something called the trapping reaction. And I will describe what we mean by the trapping reaction, but this is the reaction that it undergoes. Glucose will be phosphorylated in an ATP-dependent fashion. Okay, this hydroxyl will have a phosphate put on it. Okay, that phosphate comes from ATP. That's why I said it's ATP-dependent. All right, and it makes or produces glucose 6-phosphate. All right, glucose 6-phosphate is going to be the first intermediate in the glycolytic pathway. It's also going to be an intermediate in glycogen synthesis pathway. Okay, it's also intermediates in gluconeogenesis and glycogen breakdown. Okay, glucose 6-phosphate is really important. All right, hexokinase is the enzyme that catalyzes this reaction. Now, real quick, why the trapping reaction? Well, or why do we call it that? And the reason is because glucose 6-phosphate, right, is not a substrate for GLUT transporters. Meaning this, the KT of GLUT transporters for glucose 6-phosphate is way larger than physiological concentrations of glucose 6-phosphate. The rate of transport of glucose 6-phosphate out of the cell by the GLUT transporter is really, really, really low. And there's nothing else that will move it out. So once that phosphate is added to this glucose, it's trapped within the cell. Unless there's some other enzyme that can remove the phosphate, a phosphatase. The liver is one of the only tissues in the body that has that phosphatase. Okay. Small intestine has it, the kidney has it. The muscle, skeletal muscle, does not have it. Cardiac muscle does not have it. Neuronal tissue does not have it. Okay, so it's trapped in those cells, okay, for the most part. Now, here's the wonderful thing about biochemistry. It used to be, like long time ago, it used to be that we would just say hexokinase. In fact, we called it glucokinase. And there was just one of them that we knew about. But now there are 4 of them. Isn't that fun for you guys? It becomes more complicated the further we get. Okay, there are what we call 4 different isozymes or isoforms of hexokinase. How many of you have heard of an isoform? Okay, let's do this then. That wasn't like an overly enthusiastic response. What is an isoform? What do I mean by that? What is an isoform? Let's make sure that we're all on the same page with that. I'll give you a minute 30. Give me some answer on that. Okay, what is an isoform? Quick answer. Okay. You can talk to each other about it too. You don't have to sit in silence. Okay. "20 seconds. Submit an answer." Okay, 5 seconds. 4, 3, 2, 1. Here we go. All right, maybe I should give you guys more time on these typing ones, but I didn't. Okay. I keep forgetting to change this. Did it say they have the same number of atoms? Is that what it said? Or I didn't see it. Okay, so Different form of the same genes, functionally similar proteins. An isoform is a different functional unit of a specific molecule or protein. The functions usually are similar but slightly different, okay? Similar type of enzyme, same atoms arranged in a different molecular structure. Similar structure with similar functions. All of you kind of hitting around. An isoform is a molecule with the same chemical composition as another, but with opposite orientation. Is left-handed? Actually, that's not true. Is similar structure, but found in different parts of the cell. Okay, not really similar proteins with different function. Yeah, that's, no, that's not really it. Different forms of an enzyme that interact with the same substrate. A molecule with the same chemical makeup configuration, no. Okay, so we're not, there's not really a consensus on this. Some of you have heard of it, but maybe you don't know what it is. Different forms of an enzyme with slightly different function. That would probably be the best that I've seen so far. Let me say this, okay? So an isoform is, actually I wrote the Encyclopedia Britannica definition down if you really want it. Any of two or more functionally similar proteins that are encoded by different genes or transcripts. Okay, that's really the best way to think about this. They're functionally similar, small differences, but they're encoded by different genes or different transcript. By different transcript, we mean that they've been alternatively spliced. Okay, so their amino acid sequences aren't identical. Okay, otherwise they'd be the same enzyme. But their function is either identical or very, very similar. Okay, so let's get back here to then hexokinase. Now, here's the reason I wanted to mention that, because we're going to run into this class. In this class, there is isoforms of, I mean, we could talk on and on and on and on and on about isoforms. We're going to talk about different isoforms. All right, hexokinases is our first example of this. There are 3 different hexokinases that we call hexokinase 1 through 3 that are virtually identical from the standpoint of their Km and their regulatory capacity. Okay, these are found in extrahepatic tissue. Okay, extrahepatic, by the way, would be outside of the liver, not expressed in the liver. Okay, these are not found in the liver. They have really low Kms, 0.01 millimolar. Okay, that's significantly lower than glucose concentrations in the cell, most cells. They are inhibited by their product, feedback inhibition. Glucose 6-phosphate will inhibit them. Okay? All right, that's hexokinase 1 through 3. We discovered, first of all, hexokinase 4, what used to be called glucokinase. It's in the literature sometimes still referred to as glucokinase. We'll call it hexokinase 4 in this class just to be up with the current nomenclature. Found pretty much in the liver and the pancreatic beta cell. That's about it. Okay, it's a K-half. It's a cooperative enzyme. Okay, for glucose is 10 millimolar compared to 0.01 millimolar. Right, that's a big difference. So kinetically, these are very, very different. Okay, all right, and it's not sensitive to glucose 6-phosphate, meaning it's not inhibited by that. Okay. All of these 4 catalyze the phosphorylation of glucose, making glucose 6-phosphate. Every single one of them catalyzes the trapping reaction. Different tissues, different kinetic properties, different regulatory properties. Same name, with the exception of a Roman numeral. Isn't biochemistry awesome? Hey, you guys signed up for it, and you're too deep in now, right? I mean, you're committed at this point in time. Okay, so we didn't tell you about all this kind of stuff in freshman seminar, but too bad. All right, now, all right, so that's going to be important, okay, especially tissue types, okay? Questions on that, isoforms, what do these things mean? Awesome, let's do this. Let's talk about the liver and the pancreatic beta cells, okay? Specifically the liver, we'll be thinking more about the liver, but this will come up when we talk about the pancreas later on, in particular mechanisms for which it releases insulin. This will be important. Okay, so first of all, okay, number 1 thing that we need to say here, right? The membrane has GLUT2. Which has a high KT. Okay, 17 millimolar, as I've already mentioned. Now here's the second thing that we need to know about the liver and the pancreas, okay? It has lots of GLUT2. In the membrane. Now, I mentioned this with enzymatic pathways. We said that reversible steps are catalyzed by enzymes that are in large quantities, or they're very efficient. What we have here, this is not a metabolic pathway, this is just to get glucose into a cell, but we have here a transporter which is like an enzyme, and there's lots of it, and it has really high KT. Okay, so what are the implications of these things? Okay, that's what we need to think about, implications. One, okay, rate of absorption increases linearly or virtually linearly with changes in glucose concentration. This is the KT, right? That was the graph I showed you. 17 millimolar, okay? Large KT, much larger than physiological concentration. That means if glucose concentrations in the blood happens to double, then the rate of these GLUT transporters will also virtually double. It's very sensitive, and that's true of the liver and the pancreas. The second thing here that we need to say is this, that the concentration of glucose inside the cell equals concentration of glucose outside of the cell. And that's because there's a whole lot of that stuff on the membrane. It equilibrates it. Okay? Now, understand what I'm saying here. The pancreas and the liver have an equal concentration of glucose on the inside of the cell as what's found in the blood. And it's very sensitive to changes in that concentration. So that if the blood concentration of glucose goes up, then the concentration of glucose in these cells will also go up at essentially the same rate. Or if it goes down, in these cells they'll also go down. That makes these two tissues, the liver and the pancreas, very sensitive to changes in glucose concentration. And these truly are detectors of glucose concentration in the blood, and very important in regulating blood glucose concentrations. Okay. Question, yes?
In this situation, as you said that they're very sensitive, is this, I guess, is there a strong correlation between these GLUT2 transporters and gluten intolerance? Or gluten, I guess gluten, not it.
Glucose intolerance?
I mean, it's insulin.
Insulin insensitivity and diabetes. So the question is there a correlation between the sensitivity of these GLUT transporters and insulin insensitivity, right? So, no. I mean, the insulin insensitivity is going to affect a number of things in terms of glucose concentration. I'm trying to think the correlation. If there is, so let me put it this way, okay? Because I think what you're asking is there a cause, right, of insulin insensitivity? And so we're going to talk about insulin insensitivity. That's usually something that we refer to more of the skeletal muscle being insensitive to insulin. Now this can, because we're talking about the pancreas, there's type 2 diabetes can actually be caused by not just insensitivity to insulin, but also no production of insulin, which can be correlated with GLUT functions. Okay, though I'm not sure it's causative. Okay, I'd have to look in the literature. Yeah. Other question?
So you mentioned that like the liver and pancreas are very sensitive because of their reference to the glucose concentration in the Does
Yep.
the glucose get into the blood through the pancreas and the liver, or something else? But like the liver and the pancreas absorb the glucose and then put it through the blood, so why would it be in the other direction?
Well, okay, so the question is in regard to that anatomical picture that I showed you. Isn't it like the glucose goes into this first? It actually goes to the liver first. It circulates all the way around, and then it's detected by the pancreas, and then the pancreas takes action. Okay, what happens is that as it circulates around after it goes to the liver, okay, during a meal, let's say you just started eating a meal, that glucose concentration in the blood being absorbed by the small intestine is going to go up. The liver's not going to really have any idea. It's going to start to absorb some glucose. When it gets around to the pancreas is going to detect that. And then it's going to excrete something, insulin, and it's going to go right to the liver. And the liver is going to then take action based off of the insulin. Okay, so in terms of detection, the pancreas is really telling the liver what to do. And it will tell the liver what to do with glucose, and we'll talk about that later on. Okay, but both of these tissues have this equilibrium between what's in the blood and the tissue. Good question. Yeah, good questions. Other questions? Let me say this, because I want to get to skeletal muscle. Rate of absorption increases linearly with glucose concentration. Okay. Also true of trapping. And by trapping, I mean the hexokinase. If you look at hexokinase's it's K half, it's 10 millimolar. It also is going to be very sensitive to changes in glucose concentration. As it goes up, the liver will start to trap more and more of the glucose. If the level of glucose concentration in the blood is very low, then the liver is not going to be trapping very much glucose, okay, using the hexokinase. Okay. Again, because the trapping reaction by this is catalyzed by a hexokinase 4 that has a very large K half. Okay. So those things work in concert. That's true of the pancreas as well. Okay. Now we're going to unfold this story. This is the start of a long story that we're going to unfold. Okay, but a very important start. Okay. Okay. Questions? Any other questions? Too bad. I'm going on. Okay. Muscle cell. Let's talk about the muscle cell because I want to finish this today, and then we'll do our group work on Monday. Okay, so the muscle cell, here's what we're going to say about this. We're going to start talking about the GLUT here, and this is where you're going to really see the contrast. GLUT4, okay, is what is in this. This KT is about 5 millimolar. Now here's the really important part. Okay, this is insulin sensitive. Oops. And you all say, wait a second, what is that? What do you mean by that? Well, I mean it's sensitive to insulin. Oh. Whew. It's a good thing I came to class today. Right? Okay. So it's insulin sensitive. What does that mean? Well, let's come here. Okay. Now, this is true of adipose tissue as well as skeletal muscle. Okay. They're the same thing. Okay. So let's look on the right here. Okay. And we're going to start here. All right? So that is an adipose cell. Again, same thing as skeletal muscle. You can see here, let me just show you this. There's these big things here. Kind of draw these. Those are fat droplets. Okay? These black circles. You'll see the green speckling. In this experiment, okay, the researchers have tagged GLUT4 with green fluorescent protein. And what is being illustrated here is that the GLUT4 is distributed throughout the cell. It's really concentrated in the middle of the cell, but it's distributed throughout the cell. This is without insulin. Okay? Now, go here. This is with insulin. The green fluorescent protein, which is covalently attached to the GLUT4, once insulin is added to this culture, you'll notice what happens to the green fluorescent protein, and therefore the GLUT4. It's all on the outside of the cell. It migrates to the membrane, okay, in response to insulin signaling. That's what I mean, it's insulin sensitive, okay? that the membrane doesn't have very much GLUT4 in it until insulin's around. And then it's got a load of it in it. Okay. And that would be true for skeletal muscle. Skeletal muscle is harder to culture than adipose tissue. So this experiment is easier in that tissue. Yes?
Is it worked out how the receptors are trapping to the surface response?
Yeah, okay, so the question is, it worked out, like, what's the mechanism? So we add insulin and something magical happens, right? So I believe that entire pathway is worked out, okay? We'll talk a little bit about insulin signaling. I don't know if we'll dive into the depths of all of it because it can become real hairy real fast. But yes, I believe so. If you were to look in literature, it would have the steps. There might be some missing, but I'm fairly certain that's the whole thing. Yeah. Okay. All right, so this is just showing, I'm going to go ahead and skip this slide, but this is just essentially showing this. It says insulin binds to the insulin receptor, activates, right? This is GLUT4 here. Yeah, it has. You have to make the noise. That's what it does. Okay, and then all right, right. So GLUT4 exists in these vesicles inside the cell until insulin's there, and then those vesicles migrate and fuse with the membrane. Okay, and then GLUT4 is there. Now, who cares, right? That's the who cares question. We got 5 minutes to answer who cares. Okay, well, let's think about it this way. Okay, all right. So that means this. Okay. Implications. One, these tissues, adipose tissue and muscle tissues, rate of absorption depends on insulin. I think that's probably a pretty obvious implication. Now, here's the next implication, though, for that, and this is why we care about this. This is why you and I care so much about something like this. Think about the amount of muscle that's on your body. Some of you are like, yeah, all right. I'm there. I'm right there with you. I think about it more than I should. That's weird. Okay, anyway. Okay, so you got muscle, you got a bunch of muscle. Now compare that to the amount of liver. And you said, I never really thought about the amount of liver that I have on my body. Well, it's right here, right? I mean, it's there, and it doesn't change. You can't work it out. It doesn't get bigger. Hopefully it doesn't get bigger, and hopefully it doesn't get too much smaller either over your adult life. It's several pounds. That's about it. But the amount of muscle that you have far outweighs that. So when we start to think about absorption of glucose, taking glucose from the blood, all right, the muscle is going to take a ton of glucose out of the blood, right? And so there better be a really good way of regulating that. Because if it's not regulated, then what's going to happen is the glucose is going to fly through the liver, okay, which is sensitive to it, but it's not going to absorb it at a real high rate. It's going to get out there into the bloodstream. It's going to start to have intervention with the muscle, and the muscle is just going to start to suck it up, and you'll go brain dead in a very short period of time. Okay, if there's a tremendous amount of GLUT4 just sitting on the membrane waiting for it. Okay, so this has to be regulated. Okay? Now that gets into the trapping reaction. Think about the trapping reaction. Glucose plus ATP goes to glucose 6-phosphate plus ADP. All right, Km or Km of this is like 0.01 millimolar, which means that at glucose concentrations in the cell, it is at like 95 to 99% of the max. Which means that once glucose gets into these cells, it's trapped. Right? It's almost instantaneously, right? It goes right to glucose 6-phosphate. Now here's the really important thing about this, though. This will feed back and inhibit that. So that, and think about this, you just ate a meal, okay? Insulin is released by the pancreas. Insulin tells the skeletal muscle, go ahead and start taking some glucose. Otherwise, we're going to get hyperglycemic real fast. It doesn't really say that, but that's the idea, right? And I'm personalizing things that don't have personality. I get it, okay? But anyway, it gives that idea. So then, right, so then a whole bunch of GLUT4 hits the membrane. Okay, these tissues start to absorb the glucose. They start to instantly convert it to glucose 6-phosphate. Now, if you're just sitting around like you're doing right now, then glucose 6-phosphate levels are just going to keep rising, and they'll feed back and they'll inhibit that process. So it'll stop trapping it. Now, because there's a bunch of GLUT4 in the membrane, it will equilibrate the concentration of glucose in the blood and the concentration of glucose in the muscle, and it will stop taking it. It will stop absorbing it. So that, again, the muscle doesn't just drain the blood of glucose and you go brain dead within minutes. Okay. So these two features, the hexokinase 1 through 3, real high Km, but the inhibition by glucose 6-phosphate, and the insulin sensitivity are very important for our physiology. Okay. Well, some other things that we'll talk about, okay, with the muscle. Interestingly, the muscle is also one of the few tissues that can store a whole lot of glucose in glycogen. Okay. So when we take off on a sprint, it doesn't really get its glucose from the blood. It gets it from itself. Breaks down glycogen. Okay. That'll be one of the quickest ways that it gets energy for running on a sprint. Okay. All right. I think that's it. Are there any questions? Yeah.
Is there any specific link transporter for brain tissue?
Yes, GLUT3. So I'll mention that later on. Yeah, I'll try and mention that maybe Wednesday next week. So, real quickly, group work on Monday, not Wednesday. So we'll do our group work on Monday. So come ready to get into groups of threes. You're gonna have to talk to each other, and you have to do a wonderful worksheet. It's gonna be lots of fun. All right. See you then. Have a good weekend. Hello.